Welcome everyone. So today we are back with our past paper session and we are going to solve October-November 2025 paper 4 variant 1. Okay. So let's start with our question number 1. It says that apple juice is made by cutting and crushing the apples. When the apple tissue is cut up and crushed, it can become a darker color and go brown. Some students investigated the effect of vitamin C on the color change in the apples when they are cut up and crushed. So the students followed the following instructions. So you can read through the instructions that they first label a large test tube and add 5 cm cube of vitamin C solution, then into another test tube 5 cm cube of distilled water, and then in the third test tube they do not add any liquid, which means they expose the apples to the air. Then they cut 3 cubes of apple, each 1 cm by 1 cm by 1 cm. Do not include any skin or core. Cut each cube into the small pieces. Take care to keep the pieces from each cube separate and add the pieces of one cube to the test tube containing vitamin C solution, then into the distilled water, and then into a test tube with no added liquid in the simply exposed to the air. Use a glass rod to gently crush the pieces of the apple in the bottom of each test tube. Clean the glass rod between using it in each test tube. This is an important point so that there is no mixing up. Okay. So let's start with the first part. Describe the most appropriate piece of apparatus to measure the required volumes of the vitamin C solution. Okay. Okay, so describe the most appropriate piece of apparatus. So it must be a syringe or measuring cylinder, right? Just a second, let me set the. Okay, so we can either use a syringe or the measuring cylinder. Why? Because they give us the range of 5 cm cube. Okay, explain why the students were told to clean the glass rod. Obviously, when they are mixing the apples with the liquid must be left onto the glass rods. Hence, they have to clean it up before putting it into the next glass tube so that the liquids do not mix. So, we have to do this in order to prevent mixing of the liquid between test tubes. Perfect. Okay, next question, next part. The students noted that apple tissue in each test tube was white when they started the timing, and they observed the color of the apple tissue in each test tube at 5, 10, and 15. At 5, 10, and 15 minutes and recorded this in their notebooks. So figure 1.1 shows one of their notebooks in which they have noted down their results. So for vitamin C, for the first 5 minutes, it remained white. Till the 45 minutes, it remained white, which means there was no color change in the test tube of vitamin C. Then in the distilled water, it has gone from white to pale yellow to dark yellow from 5 to 45 minutes. And with no added liquid, it quickly goes from yellow to dark yellow and to brown. Very easy results to be interpreted. Okay. So first part asks you to complete the headings in table 1.1. So you can see that this column is for time. And we also need to write the units, time in minutes. And these 3 columns are for color change in apples. So this is the color change. Now we have to complete the table. You just need to copy down the results of the student. I'm sorry. Okay, so for vitamin C, it remained white throughout. Let me zoom in a bit. White for all. Then for distilled water, it was for the first 5 minutes, it was white, and then pale yellow, then dark yellow. Okay, for no added liquid, it was white. Then it was for the first 5 minutes we had yellow, I guess. Yeah, yellow, dark yellow, and brown. Yeah, yellow, dark yellow, and brown. Okay, now coming to the next part, using your observations in table 1.1, state what could be concluded about the effect of vitamin C on the color change when the apple is crushed and cut up and crushed. So you can see that the vitamin C showed no change in color, right? So we can write that vitamin C prevents change in color. Okay. Next part, using your observations in table 1.1, state what can be concluded about the effect of the distilled water on the color change when the apple is cut up and crushed. So we can say this you could see in the table that for distilled water, for the first 5 minutes, it remained white, then it becomes light yellow and then dark yellow. However, comparison to no added liquid, you could see that for the first 5 minutes, apple quickly became yellow, dark yellow, and then brown. It means that distilled water is slowing down the process of change in color, right? So we can write that distilled water slows down the change in color as and it was not as effective as vitamin C. Okay, now the next part. Students suggest the students were aware that there may have been sources of error in the method used which affected their observation. So there are several sources of error in this experiment. First could be because see they have not mentioned the use of color chart in order to distinguish the color differences, right? So we can mention that it is difficult. We can use or a color chart in order to describe the colors. Secondly, what could else be the source of error? For example, the cubes are not cut up at this for the same degree, or they are not crushed up to the same degree. Because see, we can say about the size. Okay, the size are same one by one by one, but are they crushed for the same amount of time? No, that they have not mentioned about it, right? So we can also mention that. The third thing is that they have not mentioned that the three test tubes apples, or you can say the apples for the three test tubes are cut at the same time. Which means what? That the first test tubes apples are exposed to the chemical for a longer period of time, then for distilled water, and then to the air. So we have to make sure that three people cut the cubes at the same time and put the apples into the test tube at the same time. So these are the few errors. So let's write one. For example, the last one we discussed is that time taken to cut and crush apples must be same for all test tubes. So, three persons could cut apples simultaneously and put in put it in test tube. Okay, so this was the error, and how can we rectified. Okay, so the student next part is the student then measured the pH of vitamin C solution and the pH of the distilled water using the universal indicator paper with the following result. The pH of the distilled water was 6 and the pH of the vitamin C solution was 4. Describe how you would use the universal indicator paper to determine the pH of the liquid. It is the most easiest part, I would say, because you know that you just need to dip the paper in the liquid and compare the color of the paper with standard color chart. Okay, now coming to the next part. When apple tissue is cut up and crushed, an enzyme comes into contact with oxygen in the air. In the presence of oxygen, the enzyme catalyzes the reaction of a colorless molecule into the cells of the apple tissue to cause the change in color. So basically, they're saying that when the apples are cut and they're exposed to the environment and to the air, there is some enzyme in the tissues and the cells of the apple which allows the catalyzation of the reaction which produces a colored pigment. Okay, so they're asking you that to explain the effect of distilled water in vitamin C. So for distilled water, you know that you observe that distilled water slows down the process of changing color. It did not stop it, which means that by putting the apples into the water, it prevents the availability of the oxygen at the same speed, right? So we can write that distilled water prevents the, I would say not prevent, it reduces. Prevents means that it did not allow it, huh? So it should be reduces. Or you can say slows down. Reduces the availability. See, these keywords matter a lot. So it reduces the availability of oxygen to the apple cubes. Okay, now for vitamin C. So you know that vitamin C it is an acid, so it must have lowered the pH. Vitamin C lowers the pH. And from the enzyme, and you know the effect of pH on the enzyme, that when the pH gets too low or too high, it can denature the enzyme. So the vitamin C lowers the pH and denatured the enzyme, which prevented the color change. Because see, when the enzyme is not working, the reaction won't occur, and when the reaction won't occur, there will be no change in color, which stops the change in color. Okay, now moving on to the next question. It says that yeast is used to produce bread. Flour is mixed with water and used yeast to produce the dough. The yeast breaks down the starch into the flour to produce sugar. The yeast uses the sugar for respiration, giving off bubbles of carbon dioxide. Simple. They are telling you about the whole process of how the dough rises due to the yeast. The bubbles of the carbon dioxide are trapped in the dough, making it increase in volume before it is baked. Some students tested the effect of yeast. Okay, so what are we measuring? We are measuring the effect of the yeast in four doughs, each made from different type of flour, which are wheat, rye, buckwheat, and maize. The effect of yeast in the dough was evaluated by measuring how much the dough increased in volume. So we are testing effect of yeast by measuring how much dough has risen. Okay. So coming back to the question, the same mass of each type of flour was mixed. So they have made sure that they are using the same mass of each type of flour, and they are also making sure they are mixing it with the same volume of water and the same mass of yeast to form a dough so that your experiment stays reliable. So they made 400 cm cube measuring cylinders were labeled and dough was added to each. The volume of the dough in each of the cylinders was measured. So basically they kneaded the dough and they put it in the measuring cylinder to measure the change in the volume. So the first part is asking figure 2.1 shows the measuring cylinder with dough made from wheat flour after 30 minutes. So record the volume of the dough in table 2.1. So the first one is approximately 38 cm cube, right? So we have to put it into the table, which is here. Okay. This this value is missing here in table 2.1. Okay. And now part 2, complete table 2.1 by calculating the total increase in volume of each dough after 45 minutes. Okay, so after 45 minutes, you have to tell the total change in volume. So this is the initial value. This is your final value. You just need to take out the difference. The first has been already done for you. For the rye flour, the difference is 11. For the buckwheat flour, 23 minus 21, it is 2. 22 minus 20, again, it is 2 here. Now, moving on to the next part, this is basically your grid. On the grid, draw a bar chart to show the total increase in volume of the four doughs after 45 minutes. Okay, so the first of all, you have to label your axis. Whenever you are given a graph, you have to, first of all, you have to think of three things. You have to label your axis, you have to decide your scale. Okay, bar graphs are a little bit tricky because the boxes are very small and it disturbs the eye to a greater extent, I would say. However, the other graphs are much easier because they have bold lines and the non-bold lines, so it is easier for you to mark them. So I'll try to make it as easy for you. Um, so first of all, let's label the axis of on the x-axis will be adding our types of flour because you need to make the bar chart. Okay, so on the x-axis, you can have your types of dough, which are four, right? And on the y-axis, you can have your total increase in volume because you have to plot these two things, you have to show the total increase in volume. So on the y-axis, we'll be having our increase in volume. All right. And you must mention the units as well on the y-axis. On the x-axis, we did not add any unit because it was a qualitative quantity. Okay, now it's very easy. You have to make the bar chart. What you need to remember while making the bar chart. Firstly, I told you that your axis should be completely labeled and that your bars should be labeled centrally. What does it mean? That whatever bar you make, for example, this, it should be mentioned as wheat flour, for example, okay? And then if you make a rye, then it should be written in the center of the bar that it is rye, okay, on the x-axis. So this is another thing that you should be careful about. Secondly, your scale should be appropriate in size. What the criteria here is that you should at least use 50% of your graph, and this is the rule for every subject, be it physics, be it chemistry. Whenever you are when you are given the graph, you have to make sure that you use at least more than 50% or at least 50% of the graph. Okay, uh, in both directions. Okay, x-axis as well as y-axis. Next important thing that you should be careful about is that you plot your values correctly and that you should use a ruler to mark your lines for the bar, and this all should be equal in width, because obviously the height won't be, the length won't be equal because of the different values, but the width of each bar should be equal, okay, and they should not be touching each other, the same protocol that you follow in mathematics for your bar graphs. Okay, so let's quickly label our y-axis. So on y-axis, I would suggest that take 5 small boxes for 10 units, which means that each small box will be equal to 2 cm cube. Now let's quickly label our diagram and plot our bar graphs. So now you can see that as I told you, that I marked five small boxes as 10 units. So each small box will be equal to 2 cm cube. Okay, my scale wasn't working appropriately. So I had to draw it with my hands, but you need to remember that you can't draw it roughly with hands. You have to use your rulers. Okay, now coming back to the next part, the students wanted to know why the effect of yeast in the 4 dose varied. Table 2.2 shows some of the biological molecules in the 4 flowers. Okay, now this can be a bit tricky, but let me explain it to you in easiest way. Okay, now you can see that wheat flour has shown us the greatest rise in the dough, right? Then buckwheat flour, and then the maize flour. And also you need to write the full forms. Okay, I have used the short forms due to the time and the space constraint. But you'll have the whole page, so you know that you have to write the full forms. The students wanted to know, okay, so basically you can see in the question, in the table, that wheat flour has the starch 54.5 grams, rye flour has the starch 54.1, buckwheat has almost near to it 59, and then maize flour 74. So it is basically in the approximate ranges. Now, when it comes to gluten, and always remember, a lot of people are confused that gluten is a like carb or something, but gluten is a protein. Okay, so gluten in wheat flour it is 8.9, in rye flour it is 3.1, in buckwheat it is 0, and in maize flour it is 0. So yeah, the people who are allergic to gluten, they use buckwheat flour and the maize flour. Okay, so you can see that there is a major difference in the content of gluten in the 4 types of flour. Now, when it comes to fiber, you can see that 10.6, 13.7, 10.4, and 4.3 maize flour for maize flour. So you can see the greatest difference in the content is in the gluten. Okay, so it is gluten which could be making the difference in the rise in the dough. So use table 2.2, your bar chart and the information about the process of the bread production. Suggest and explain how these biological molecules affect the total increase in the volume of the bread and dough. Why I decided gluten? Because gluten showed the greatest variation in the amount uh, which is present in the different types of flours. So when it comes to gluten, it could have made an effect because you can see that gluten was greatest in wheat flour, and it shows the great and the wheat flour also showed the greatest rise in the dough. Um, However, the wheat flour, the rice flour, and the buckwheat flour, they have approximate values of starch and fiber. So we can say that they had the little effect. I'm not saying that it had no effect, but it could have the little effect on the differences in the rise and volume. Okay, so let's start writing our answer so that it makes more sense to you. You have to write an answer for this P mark. So first of all, what we have concluded that gluten has an effect as it matches as it matches with the rise in dough. And second thing, we found out that starch or fiber have little effect as they do not match with the pattern. Okay, what else you could write is that what was the pattern that you were seeing? Is that large volume increase in dough, increase in dough is due to trapping of air bubbles. Okay, now moving on to the next part, describe, one second, describe how you would show that each of the flours contain starch. So basically they are asking you about the test for the starch. So it's very easy. You have to add the iodine solution, which will turn blue or black. So add iodine. It will turn the flour solution or flour blue black. Okay, now coming on to the next part. The students made more dough from wheat flour, yeast, and water. Using the dough, plan an investigation based on the method in 2A. They will be following the same method which was used in part A. To determine the effect of temperature on increase in the volume of the dough at temperatures between 20 and 80 degrees. You have to experiment, you have to do the same experiment, but with little or slight change, which is basically that you have to measure the effect of what measure effect of temperature between 20 and 80 degrees Celsius specifically. So obviously first you will make the dough and then divide them equally. So I'll write the points simultaneously, so you can decide whatever points you would like to write. So first of all, you will make the dough and divide the dough into equal parts Okay, that should be that could be your first point. And at least you should divide your dough into three into I would write it here in in three cylinders. Okay, three measuring cylinders at least. Okay, next point is since you have a range of temperature, so you should at least take three different temperatures, right? So take three different temperatures. All right. And how could you change the temperatures? You can use the water bath. This is the most used method of changing the temperature. Sorry, I'm writing it in a bit hurry, so my excuse the writing. Use water baths. Okay. What else you would see that you'll have to measure the change in volume, right? Measure volume change at regular intervals, regular or same intervals that you have decided for your experiment. Okay. Um, Sorry. So now you have to calculate at the end, what did you do in the first experiment? You calculated rise in volume after 45 minutes, which was your standard. First, you measure it at regular intervals, and at last, at the 45 minutes to measure the actual change from initial to final. Okay, again, this is the repetitive thing, which is that you have to replicate your experiment. So replicate your measurements and take the mean value in order to avoid the chances of errors. And at last, what you did, you plot the graph. So see, they have not given you an extra thing. You have to just copy down the same experiment with some changes in it according to the given scenario. What could be your expected results? Obviously, if you are increasing the temperature, the enzymes would work quickly because they were good at optimum temperatures, and hence your volume will rise quickly at the higher temperatures. So, what are our expected results? That volume increases as the temperature increases. This is an expected result. But you also know that the enzyme shows the optimum activity at the optimum temperature, and then it slows down and stops increasing eventually. Now coming to the last question of this paper. Starting with question number 3, it says that a student investigated a plant that had some stems with flowers and some stems with no flowers. The student picked one leaf from a stem with flowers and one leaf from a stem with no flowers. Figure 3.1 shows the two leaves, one from the stem with the flowers and one from the stem with no flowers. So in part 1, they're asking to complete table 3.1 to compare the size and the shape of the leaf. So let's quickly discuss the size and the shape and then we'll write it. Okay. So the first leaf, which is from the stem with flowers, you can say that its size is larger as compared to the second one. So for the size, we can say that the first flower is larger and the other one is smaller. Okay, you just need to tell the differences. Okay, now for the shape, you can see that the first leaf is oval in shape, approximately oval in shape with a pointy edge, right? So you just have to describe the shape. So we can write that it is oval with pointed edge or pointed tip, not edge, tip. Okay, the next one, oops, sorry. The second flower is basically wide at the base and uh, longer or pointed at the tip. So we can say that wide at the base see these type of questions are very easy and you can easily gain marks out of these questions. Wide at the base and pointed corners. So we had corners in it. Now, coming on to the next part, suggest what the student needs to do to be certain that two leaves they picked are representative of the leaves on the two types of the stem. So you know that in order to, in any experiment, in order to prove the representation or the reliability of the experiment, you have to repeat the experiment. So in this, you will be collecting more leaves. So you need to collect more leaves from both stems and check for the similarity with the initially selected leaves. Okay, that's it. The last part says that in space below, make a large drawing of the leaf from the stem with no flowers as it appears in figure 3.1. See, I told you this question is very is the one in which you can gain the marks easily. If you do not know anything, still you can do it because this does not require any of the theoretical or the practical knowledge. You just have to see and write. Okay, so I've already drawn the picture. Basically, I'll tell you the tips of drawing in this part. You just had to copy paste the diagram that you saw in the picture. Okay, but what you need to make sure that your drawing is bigger. You are using a sharp pencil. There is no breakage of lines. What does it mean? That you can't draw it like this. Okay. The lines should be joined together. The lines should not be broken. There should be the continuous lines and your pencil should be sharpened. Okay. You are not allowed to shade anything. So you don't need to see. I have not colored it. It was a black and white diagram. It appeared shaded, but you are not allowed to shade anything. Okay, there should be no overlapping lines. There should be no overlapping text and the lines. Secondly, for example, you can see in the picture, can you see that I have created deliberately the deep notches? Can you see these? This, and this, right? Because it was given in the picture very clearly that these are the notches in the leaves and these are the pointed edges. This is the stem of the leaf. Okay, so you need to be careful with these things. All right, and you also should show the branches as well because I saw I was checking this question for a student and she did not draw the branches, so I had to deduct the mark because it is given in the mark scheme that you also need to draw the branches as well. And it is very clear in the picture that you can see the branches. And you all have drawn the leaf in your kindergarten, for God's sake. So you know how to draw the branches as well. Okay, so whatever you are seeing in the picture, you have to copy it with a bigger size. If you draw it small, like this tiny size, you won't be getting any mark. All right, so you need to draw at least a bigger diagram that at least the size same as the picture. You can make it a bit bigger, but at least try to use the space as much as possible. Okay, so this marks the end of the question here. Okay, I shall see you in the next session. Good luck, guys.